All right, it's a great pleasure to be here at this fantastic symposium today and to give a talk about our work. And I thought about, I want to talk about recent work. And this is in various subjects. And I actually gave a pitch of sort of that we can use nanotechnology. We heard already this afternoon so many tools that we can use. And those are super interesting to resolve things at a molecular scale. And as David already said, I mean, I came from solid state physics and electronic transport, and I changed my field to biology, coming from this molecular electronics and now studying the live world. And that actually is just my all-time fascination. But early on, as a sort of teenager, I chose for physics rather than biology, because it's biology, the classical view as a high school implanted into my brain is this, yeah, it's zoology. That's fascinating too, but it's very phenomenological and not so quantitative. And as a physicist, I really liked the physics more for that aspect. But that changed over time. And I think that's also partly due to technology. Now, there's, of course, a famous history of technology contributing to science, opening new worlds. We're here at the MRC. There's so many cryo-EMs, and they really show a new world in ways that we couldn't do like 20 years ago, right? Well, I'm from Delft. This is Antoni van Leeuwenhoek, already sort of like 3 centuries ago, developed an instrument. This is his microscope. Oh, excuse me about that. Sorry about that. Yeah, this microscope. Wrong button here. Yeah. This one here. And that allowed him to see a new world of small animals, as he called it, right? And he discovered bacteria 3 centuries ago. So that's a new world, and that gave rise to the field of microbiology. And here we have another Delft guy, Martinus Beiring. You may not have heard of him, but he discovered tobacco mosaic viruses. The first virus, really isolated, showing that it's a small organism, a biological entity smaller than bacteria. So that opened up microbiology, and we're very used to that term microbiology. But I think we now entered the era of nanobiology, and that may sound like a high term to you, but I mean, I think it's very appropriate, because nanoscale is the scale of the size of proteins and all that basically constitute the molecular world of our cells. Now, nanotechnology is the instruments that we can use to study and manipulate that nanoscale biological world. This is some wonderful example from Don Eigler from the 90s, when the first nanotechnology, the single molecule, single atom tools were developed. This, what you see here, actually every dot is a metal atom, a noble gas atom on a metal surface here. And what Don Eigler could do is to do scanning probe microscope and take one of those things and put it at a surface. And so build a quarter, half, 3/4, full circle of these atoms on a surface. And then you see sort of a metal interference wave appearing at the metal surface. Well, it's just an example of nanotechnology, because it shows the ability to at the level of single atoms and molecules, have the tools to image there, to build there, and to measure novel properties, because this property wasn't there first, and now you can actually use these single entities to study these properties. So for me, nanotechnology is a toolbox that we can now also employ in biology. And that's what we've been doing in the last, in the whole community, John and many other people, doing in the last decades. So in my talk, I will give you 3 examples where you show that imaging is very powerful. That's not much of a surprise, because many of us, almost all of us, will use microscopes. But I'm still myself amazed that if you're at a microscope, you see a single molecule blinking. I'm this person of 2 meters, and there's something happening on the 2 nanometer scale that I can actually see. So I find that still amazing. You can measure quantitatively on proteins, and we can also start building with these tools. So that's the theme of this talk. So I show you some particular research examples of the power of this nanotechnology to do some biology. And the first topic revolves all about genome structure, which already is a very old subject. You see here these wonderful pictures from Walter Fleming, microscopy pictures, 1882. That's a long time ago, 1.5 centuries. So we're studying chromosome structure already for a very long time. And still we have many questions, and still there's many recent developments in understanding chromosome structure. You see actually interphase, and then towards mitosis, you see basically the famous mitotic spindle separating the two, the daughter chromatids. Our current view is actually that in the nucleus of the cell, every chromosome occupies a certain territory, and if you zoom in, there's compartments, and if you zoom in, the basic motif of our chromosome polymer wire is actually loops, okay? And if you zoom in at the stem of these loops, you often find a complex, which is so-called, goes by the name SMC complexes, goes by the name like condensin, cohesin, SMC5-6. These SMC complexes are the star of this first part of the talk. SMC stands for structural maintenance of chromosome, for proteins, and they're very large, 50 nanometer, 50 nanometer large. They cold call here, a cold call protein here, and then this intrinsically disordered glycine, connecting it, making it basically a circularly connected protein complex with a couple of subunits there. And these play the major role in orchestrating and regulating our chromosomes. And these are very well conserved, bacteria to human. And basically, the first part of this talk is about this excitement that we discovered that there's a process called DNA loop extrusion. So, as I told you, there's the stem of the loops are these protein complexes, and this simple cartoon, artist impression, of course, and I'll show you some real data in a minute, is basically showing the idea that there's a protein complex landing on a DNA and it's starting to make a loop. And that is basically of major importance in many aspects. So these different families of SMC proteins, condensin, cohesin, SMC5-6, are involved in anything, in mitosis, in condensing your chromosomes, in the centromere definition, in DNA repair, in gene regulation, long-distance gene regulation, recombination in meiosis, and viral silencing. And actually, they're not just academic interest. Actually, there's very severe diseases. If you have cohesin deficiencies, you get a Cornelia de Lange syndrome, for example, like this girl, or they're involved in certain cancers and all that. So these are very important proteins that are there, and we just recently discovered they're actually molecular motors. It's I find very special. Okay, so I came into this field some 10 years ago or so, and then there was a dispute in the field that it was clear that these SMC proteins were in some way connected to condensing our chromosomes, for example. Now, if you take, and the common view was that if you have a polymer wire, you big DNA, you want to condense it, you can do, for example, that you have a protein binding and it bumps into something else, it clicks there, it clicks here, it clicks here. You just clump the whole thing together. That is a way to condense your chromosomes, right? And it was thought to be like that because these are ATPases, these SMC complexes, but fairly lousy ATPases. They burn only a few ATP per second. There was another hypothesis called DNA loop extrusion, where the idea is very different, namely the following, that you clamp such a thing and then you extrude a loop of DNA and another one and so forth. And this is a very efficient and elegant way to also condense your chromosome. This actually, if you trace down the history, the idea of DNA loops goes back to 1983 even, where people proposed this, and Riggs and Naismith proposed this. But it was only sort of until 2016 or so that people started to come up with this more particular hypothesis that this might be a way. But this, and if so, this could be very elegantly explaining, for example, the formation of mitotic chromosomes. Because if you have your own self, you only basically condense your own DNA and not a replicated copy of the DNA. So it would automatically lead to this kind of self-assembly of mitotic chromosomes. But these are polymer simulations, and it was still very controversial. So then we came with all our single molecule techniques. So visualization with scanning probe microscopy, visualization optically with, for example, DNA curtains and all that. And John already explained to you magnetic tweezers, where you have a bead. If you throw in these SMCs as a function of time, you can see the condensation process in real time. So these kind of single molecule techniques really were helpful. The nanotechnology here was helpful to resemble this. And maybe the most striking essay was in the end a very simple one, where we used turf near-turf, high-low microscopy to visualize a DNA molecule on a surface. So here we take a very long DNA molecule, say lambda DNA, 50 kilobases or so, and we attach it at the two ends on a surface, and we put a intercalator dye for visualization. And then you get a picture in your microscope like that. Here you image the DNA molecule. Now what you can do is to throw in condensin SMC, motor proteins there, and ATP. Oh, the movie doesn't work, I don't know. And you see a cluster there of that stuff appearing. And if you now apply a flow in the plane of these molecules, you see that this is not just a cluster, but it's actually a loop. In fact, we could see in real time before your eyes the formation of such a loop. This was work by a postdoc, Maiba Ganji, collaboration with Christian Hering's lab, who purified these proteins in Heidelberg. And here you see a DNA molecule attached to the two ends. There's a flow pushing it a little bit up. And now there's actually a loop growing here as a function of time on the time scale of a minute or so. You see this loop growing as a function of time. So this was unambiguous experimental evidence. Yes, this loop extrusion occurs. There's really a motor at the base of these loops that makes these loops, that extrudes these loops over time. And then of course, lots of biophysics questions could be answered. For example, the first one is that they found these are very asymmetric loop extruders, so they don't extrude loops like that, but more like they bind it and extrude from one side, the loops, for example. You could see that, of course, they needed ATP, fuel there. They have a remarkable rate. They can extrude these loops. This is a kilobase per second. Okay, that's a lot, right? I mean, we're used to polymerases that make a step every few tenths of milliseconds of a base. Here we talk about in a second a kilobase of DNA that's transferred. On the other hand, there's a very low stalling force of a fraction of a piconewton only. So they're very fast, but very weak motors. And associated with that, they probably make very large steps of hundreds of base pairs per step and many tens of nanometers per step, which indeed later on we quantified in actual biophysical measurements. So summing up this earlier result, what we found is that there's a novel type of molecular motor and that we didn't recognize. I find it already astonishing, right? We're talking 40 years after the discovery of kinesin motors and all that. We discover a motor that's processing our DNA, and it's absolutely important for our chromosome structure and regulation. And so it's very exciting. So we have this novel motor. And in the last few years, we've been sort of now expanding all kind of measurements on what happens when two motors run into each other. At high concentrations, they make phase condensates. We measure the step size, and we look at supercoiling effects. Okay, I'm going to show you a few recent results of this long list of things that we did since then. For example, we asked this question, what happens if we don't do these experiments merely on bare DNA, but we fill up the DNA with nucleosomes or other binding proteins? Because actual DNA is, of course, is fully covered with all kind of binding proteins in our cells. So this work by a postdoc Prajan in our lab. So the first question we asked is, like, what happens? Suppose we have such a loop extruding motor, and it runs into an object which is bound on the DNA, like a binding protein, CRISPR-Cas or a nucleosome or whatever. What will happen? Will it go off? Will it block the loop extrusion? That's the end of the experiment. Will it proceed, put it basically into the loop? What will happen with some such encounters? Well, here you see a couple of experiments. Here you see an experiment. This is the same type of experiment. We visualize the DNA with some bound proteins on there, like you see here. And now we see a loop appearing. As a function of time, the loop proceeds, runs into this roadblock here. This is a CRISPR-Cas in this particular case. And now you see that after a few seconds, actually it's gone into the loop. So you see here a case clearly where the object is bypassed. It's simply put into the loop, right? Here you see another example of blocking, right? Here we see basically the same thing. A loop is starting and it runs into this object, runs here, and then basically over 6 minutes, it's staying there. It's blocked. Okay, so we do experiments like that. We have an assay where now actually we don't use so much the actual visualization because of the low stall force that is actually messing up our quantitation a bit. So we use this assay where we have a very floppy DNA and we measure the mean square displacement of this object. If it's very floppy, this is large. If it starts making this loop, it's basically tensing up the DNA. Therefore, the mean square displacement is going smaller. And later on, if it goes into the loop, it fluctuates again a lot. So we see basically quite accurately if it does indeed get incorporated into that loop. So we have different categories of experiments, ones that get blocked, ones that are passing, and so forth, and we can distinguish this. So what's the results? Now we looked at nucleosomes, RNA polymerases, DCAS9, and all of these objects basically zip into that loop. They're not blocked, they're hardly blocked by these kind of objects. Okay, we got curious, we make larger objects. For example, we had particles attached to a CRISPR-Cas, which was bound to a DNA. You can do the same experiments. So here's some experiments with some 30 nanometer gold particles. Basically, you do these experiments, and what we see again, they can basically into the loop very easily. Even larger, ridiculously large, 200 nanometer particles, right? So what is the result for the 200 nanometer particles? This loop extruding condensing is running into it, and it still simply passes it into the loop. Right? What's happening what's happening upon the encounter is the complex opening up and grabbing behind. For example, to test that, we teamed up with Jan-Michael Peters' group from Vienna, who has a particular cohesion variant, which is one big fusion protein. So here the cold-col, the glycine and the cold-col are three separate molecules. This is one big molecule which is also covalently attached near the hinge. Even that did loop extrude, and even that did pass the particles into the loop. So this made us conclude that actually counter to the common wisdom at that time is actually that there is, I'm missing a slide here, I think. Yeah, what happened? Anyway, now, okay, I can tell you this thing, sorry about that. Is that actually there's a non-topological mechanism. It's not topologically embraced. So the DNA could embrace the complex could embrace the DNA topologically or pseudo-topologically. What I showed you here, this is a pseudo-topological encapsulament of the DNA, or non-topologically. What I mean is that maybe the DNA folds itself around the DNA. But actually, if you would unwrap it, it would not be topologically bound. And indeed, we conclude this actually these measurements indicate a non-topological way to extrude those loops. Then we sat together with a couple of people. You see Christian Heering, Benjamin Rowland, Jan-Michael Peters, and myself here in this picture at the conference in TTC. And we phrased a hypothesis for a model, which we call the real and SIL model, and you see that on the screen here. And this combines all the experimental evidence at that point 2 years ago that we had, which is lots of work from Jan Leuwe and other people on the crystal structures, which resolve the particular, say, ATPase head domains and so forth, the hinge domains and so forth, Fred experiments from Jan Michiel, and our visualization experiments and so forth. And a picture emerged that it might actually work like this particular model. It looks a bit complicated, but let me talk you through this. There's this complicated greenish thing, that's the SMC complex, and that holds the DNA at this point and at this point, at 2 points. And this actually is the loop that's being extruded, okay? Now, the most important event in the whole ATP hydrolysis cycle is the binding of ATP. If that happens, basically, these two heads clamp together and they open up a binding interface for the DNA, which is clamped onto the top of these heads. Completely consistent with Jan Leuwe's and many others' cryo-UN structure. This is an important event because this DNA is double-strand DNA. It's pretty stiff. It's temporarily pushed into the lumen of this upper cold regime. And therefore, it actually opens up this structure. It pushes that hinge completely up. Now, at this point in time, ATP hydrolysis can take place. If that happens, this opens up again. Now this DNA therefore is released into the loop, and bound by the hinge there, that piece of DNA is bent back and brought back to with the NIP PBL to bind at this particular point, and you're back at the starting position, and you've created a bit of DNA into the loop. Actually, it's visualized in this movie that you see here. Maybe you have a quick look at that. So you have the ATP binding, that makes a conformation change, that pushes the DNA inside the lumen there, pushing this stuff up there, and then you get this kind of arrangement. Now this hydrolyzes, and therefore this is transferred and it folds back into its structure. And this is a way that you can imagine that loop extrusion is proceeding. And it's compatible with many experimental data, including the fact that you would have non-topological procession of this loop extrusion process. So, yeah, this was the slide that was missing. These experiments showed that there's a non-topological modus for loop extrusion. It also actually, the data show actually that loop extrusion can happen in cells, because many biological objects can simply be bypassed. So this loop extrusion can actually proceed there. So the conclusion from these experiments is that virtually nothing can stop this loop extrusion process. However, there are a few exceptions. One important one is a protein called CTCF. That's a protein that can bind on the DNA, and it was known already from high-C studies that it plays an important role in defining the domain structure in chromosomes, in interphase. So the idea is that there might be an interaction of cohesion to this CTCF, and if the cohesion does loop extrusion and it encounters that CTCF, it stops at that particular point. This is what we tested, collaboration with Jan-Michael Peters, a paper in Nature 2 years ago, where we tested this with a single molecule method. And here you see some of the data. So we labeled the DNA, we labeled the CTF protein, and we studied basically the encounters of this, the CTCF and the loop extruding cohesion. And we basically count then how often does it stop there, get blocked, or actually passes it by. And we found actually it's an asymmetric blocker. It blocks from one particular terminal of the protein more than from the other, 3-4 times more efficiently. And it blocks it actually from the N-terminal side pretty efficiently, 50%. Before I showed you most things block only 10%, but this is now 50%. So this is actually important protein in defining chromosome structure, domain structure. And a surprising thing we found is that this interaction is not like a yes-no answer, block, pass. It's actually force dependent. So depending on the tension in the DNA, this can actually, CTF can actually be a very efficient blocker, like 100% blocking, or actually completely lousy blocker if there's no tension at all. If there's no tension at all, it does not block. It's only when there's tension, strong tension on the DNA, or strong tension actually, tension on the DNA, that it blocks. Which actually is an important realization because this makes it context dependent in the cells. Because in some areas in your nucleus, there will be processes happening like transcription or other processes that actually create local tension. And then the CTCF has a different effect than if there's not the process happening. So it becomes context dependent. Here's another exception, and it has to do with telomeres. This is work with Stéphane Marquand from Paris, done by Brian Alec and Alice Desheis students, and they looked at the effect, what happens when condensin runs into a Rep1 array. John already told you about telomeres. This is the yeast telomere, which basically is an array of Rep1 proteins. And we wanted to check out what happens there. So what we did is the same type of assay. We have our assay of a DNA molecule on the surface, and actually now somewhere in the middle of that DNA, we engineer a small array of Rep1. Okay, and then we do the same type of thing. We count if it blocks or passes that particular array. Now, if we just have a single Rep1 dimer, it doesn't block at all. Fine. That's very much in line with what I told you. Small proteins don't hinder the loop extrusion. However, if we have a 100 nanometer long dense array, it blocks basically the vast majority of the loop extrusion. Right? So telomeres are not passed by the condensin, and actually biologically it makes sense. This is really a biophysical effect. It's only the DNA binding part of the protein, you say the same type of blockage, suggesting it's really the accessibility of the DNA in that array, where the condensing has to grab somewhere and it cannot do it now. If we make gaps in the array, like there, so gaps of say 16 base pairs or so, then actually the blocking efficiency is dropped completely. If we fill up the gaps with leg I, completely different protein, we're back. And if you do only leg I, there was a suggestion of the referee, we worked hard, actually same thing. So it's really a biophysical effect. There's no biochemical interaction. With CTCF, there's a clear biochemical interaction. Here's a biophysical effect. The linear dense array will basically block this kind of stuff as well. Now you can do all the things like make the array short or long. You get the predicted things. Long array block a lot, short arrays not so much. Same thing if the gap size, if you have a large gap size, you get not much blocking, and a very tiny gap size, you have lots of blocking. Again, this is all force dependent and all that. There's also beautiful in-cell experiments from the group of Stefan Macron. He did, for example, engineer such an array in the middle of a chromosome 7. And then you did ChIP-seq probing of one of the subunits of condensin. And then this red bar, for example, shows you that there's lots of condensin just right next to that array, which is engineered. However, if you now make a sparse array, in the same array you make some gaps, then again, you don't see that condensin there accumulating near that array. And this is one of the cell experiments. He has different ones. For example, if you look at chromosome proximity of two fluorophores near an array that's different if there's condensin there or not. So it's a beautiful example where in vitro and in vivo experiments show exactly the same thing. Right. So these SMC proteins are a new class of molecular motor proteins with very interesting biophysical properties. And just yesterday night, I threw in a couple more slides on the biophysical properties because I'm very excited about it. And I thought John would also already introduce these magnetic tweezers for force spectroscopy. And indeed, obviously, if you have a magnetic tweezer like that, like a bead, a DNA in a glass slide, if you throw in that stuff, you make that loop, the bead will go down. Now, you can use that for force spectroscopy to measure the step size, for example. And indeed, if you zoom in at very low force with the right conditions on the magnetic tweezer, you can actually resolve the steps. And indeed, we can measure the step size, which goes like 50 nanometers and goes down with higher forces, which corresponds to hundreds of base pairs every step. So every step that it makes, it puts hundreds of base pairs into that loop. Now, interestingly, we developed a variant of the assay, worked by Richard Janissen and Roman Barth, also collaboration with Jan-Michael Peters, where we measure that not only does these motors step like that, they also twist the DNA like that. Every step they put in supercoiling into the loop. How do we measure that? Now, that goes as follows. If you can look at this assay a little bit. So, we can take these external magnets and rotate these magnets. This DNA is torsionally constrained. So if you rotate the external magnets, the magnetic bead will follow. And therefore you put some coils into the DNA, supercoiling. This is a well-known effect by magnetic tweezers, already known for 25 years, right? And then you can make, for example, a curve of the height, where you twist on the right or twist to the left, and then the height goes down. This is this head curve, magnetic tweezers. Okay. Now, if we do our experiment and we throw in the SMC and ATP, of course, and suppose for the sake of argument for the moment that it will indeed do what I just told you, it will put twist in the loop. Then, without turning in any magnets, actually it will spontaneously put twist in the loop here, which is extruded, but it's topologically constrained. So if you do that, it actually has the reverse sign twist in the rest of the DNA molecule. Now what we can do is take the external magnets again and rotate those away, right? Just by rotation you remove those, because this is one single object which is locked there. And therefore we actually get a new head curve which is actually displaced. And displacement is the direct measurement of the amount of coils that are put into the loop. Okay, so we make a number of steps and now find that we have a displaced head curve, and therefore we have directly measured the amount of twist that's generated. Now we can count the number of steps and we can count how much twist is generated. That's what we did. Suppose you do a single step, then you can find that the amount of twist is about minus 0.6 supercoil. So next to putting DNA into the loop, it also coils it by minus 0.6 supercoil. If you have, say, 3 steps, you say, get 3 times that is 1.8. Right? So on average per step, we find very consistently that we have minus 0.6 supercoil induced in this process. This was first for cohesion. We can measure these properties. Interestingly, the amount of supercoils is the same if we induce this with a small step, so we do this, or with a large step. Suppose we make a very large step. The amount of supercoils is the same. Well, that's already very special. And also, it's actually very independent of force, whereas most other properties as step size are very dependent on force. So, we actually, this is a very conserved property happening for everything, human cohesion, yeast condensing, yeast SMC56, all minus 0.6. And so we already have, I think, to adapt our model for the real and seal, which I told you, which is complicated wire diagrams, putting it up. There's some evidence actually that these coiled coils actually make a rotation of 180 degrees. We think there's 2 contributions to that 0.6 supercoil. One is the fact that at the moment of ATP binding, you have a conformational change of that NAPBL, and actually that already induces a rotation of about 60 degrees there. On top of that, you have 180 degrees, sort of because these arms are not just stretching out, but they're also coiling about like that. And if you combine that, you get ballpark something already of the right order of magnitude. So we think that can be accounted for. So summing up, in this first part of the talk, which was also the longest, don't worry about it, I showed you, which I find very exciting, a novel type of molecular motor. And most of the discoveries there were made by just visualization. Visualization of the DNA and checking what happens there. And we found that they can pass huge roadblock, that they have a non-topological way to loop extrude, that linear proteins arrays and CTCF really stall it, and that there's interesting properties such as the supercoiling, also supercoiling. And they must have huge effects, right? Because that goes two ways. If these SMC motors put supercoils in the loops, that's interesting for the loops, but also for the rest of the DNA, because as much supercoil they put here, the reverse sign is in the rest of the DNA. So this already makes a new modus of interaction with transcription and other processes, for example. All right, let's switch gears a bit. I was telling you about using nanotechnology for different biological questions. So the second one is basically, can we measure on single proteins with nanopores? This is an old dream of nanotechnology to, for example, make a small hole and then traverse a DNA molecule through that for DNA sequencing. The idea of the experiment is really very simple. So you have a small hole, you put that in a membrane in an aquarium, you fill the aquarium with some salty water, you put a voltage on, and then there's, of course, an ion current running through the hole. Now, if the DNA, which is a negative molecule, it goes from minus to plus, if it's transferred in a linear way through there, it will actually temporarily block the ion current. If you stick it in, there's less ions going there, and you can, it's just a sensor. It's just a sensor for a molecule. It's the Coulter counter from the 1950s, but then at a molecular scale. Now, this super simple idea, which was around already 25, 35 years ago, works. You have these sensors, you can build this, and nowadays even sort of like been developed by Oxford Nanopore Technologies into a commercial device in other companies, where indeed you pass it through a biological pore, for example, and you read off ion current signals, and it's so sensitive that tiny difference between the different bases and combinations of bases give you a signal that you can actually translate into a sequencing signal. So that's DNA sequencing, which was solved sort of 10 years ago. Since then, of course, the field is moving on, and in my lab, we're basically using these nanopores now only for sort of proteins. So we still developed a couple of novel nanotechnologies, make rotors on nanopores and so on. I won't talk about that. We also work on nuclear pore complexes. But let me today talk about protein sequencing. Can we use this for protein sequencing? Not DNA sequencing, but the sequencing of proteins. Well, that's very important because you might think, you know, the human genome, we have 20,000 genes. So what is there to know? We know proteins, right? And protein coding genes, and that's it. But no, there's lots of transcriptional variants. So lots of variants already at the transcriptome level. But at the proteome level, there's millions of proteins variants. Millions, really, not 20,000, but millions. So you basically would like to know what is in your cells. You want to have tools to actually measure what's there. Right. So there are techniques, Edmond degradation, mass spec. These are there, and they do work to an extent. They're huge machines, they're very expensive, and they have their own limitations, such as low abundance proteins are difficult to measure in a sea of other proteins, things like that. So there's a dream in the field to develop single molecule detectors, and that can be done with fluorescence techniques or nanopores. I will talk here about nanopores. Well, then you have to realize, if we start a project yet, it's really challenging because they're mixed charged. There's 20 amino acids, not 4 bases. There's hydrophobic, hydrophilic. They're folded. You don't have a PCR like in DNA sequencing. You just cannot amplify a single protein to lots of copies or so. Anyway, we got started, and there's work from a postdoc Henry Brinker in my lab, and there we developed this approach, and that works quite nice. So basically, we hijack DNA sequencing here. So in DNA sequencing, you basically pull a DNA molecule through a nanopore. You don't do that spontaneously driven by a field, because that would be much too fast. Because then you need a minimal voltage to get it in, but once it's there, it zips through. So every base spends only less than a microsecond in the nanopore. That's too fast to actually get reliable information. So you have to slow it down. And the trick to do that is to take a polymerase or a helicase that actually binds to the DNA and makes a slow step. Every 10 or 100 milliseconds, you have it make a step, and then it steps this part of the DNA through the nanopore, right? This steps of about 0.3 nanometers, 3 angstroms, and that's because this particular helicase 308 makes 2 steps per ATP hydrolysis cycle, stepping the thing nicely too. So the trick we did is to take this principle of, well-proven principle, well-developed since a decade of nanopore sequencing for DNA, and now we attach a peptide chain at the end of the DNA. If you do so, the helicase keeps stepping up the DNA, and now it's putting the peptide through the nanopore. Idea is simple, right? So you can do that, and it works. Okay, so you have here, for example, a typical trace. This is an ion current as a function of time. So all these levels correspond to pieces of DNA in there. So this is actually a DNA sequence signal. This is very well understood. Here we enter the linker and then the peptide, where we also see steppy signals, suggesting that the approach works. We can actually transform the time, the actual time, to helicase step numbers and compare the DNA sequencing part to calculations, because this is well developed. So until here we understand the signal even quantitatively with good modeling of the signal, and here we have to develop our own codes to do peptide sequencing. Okay. The first thing we did is actually to check the sensitivity. For example, by substituting at one particular point in the peptide 3 different amino acids and seeing then consensus signals in red, yellow, and blue here. You see that even a single amino acid chain substitution makes a quite different current trace there, showing the extreme sensitivity. These nanopores are super sensitive. Local atomic chains can be easily measured here. And then we can do a so-called confusion matrix, which scores how accurately you can predict whether a signal is in the blue or yellow or red category, and get numbers of sort of 90%. Moreover, we found a trick actually, that if we use high helicase concentrations, we can reread the same molecule. The very same molecule. We first transferred it, and then we do it again and again. How does that work? Basically, we have a helicase pulling up the DNA peptide construct. If it reaches the peptide, it's a DNA helicase, so it cannot walk on the peptide, it falls off. But if in the meantime a new helicase that's basically docked on the back of this helicase, this will fall off, but now we are back at the situation, it will zip in through the voltage, like here, and start it again. So we can reread, not only once, we can reread tens or hundreds of times. This makes the single read accuracy super high, single molecule read accuracy. Because you can reread and get the reading areas, errors, you can take them out. All right, so we extended this approach actually not only to measure sort of proteins, peptide fragments, but to actually look at particular PTMs, so post-translational modifications. So proteins can, of course, be modified by chemical groups, and most proteins in our cells actually are. You see here a list of the sort of abundancy of phosphorylation, acetylation, glycosylation, amylation, and so forth. On the log scale here, so phosphorylation is by far the most important ones, but there's many tens of different possible PTMs, and you want to see if this technique can actually measure that. And here it is, work by Innova in my lab. It's basically compared, say, a peptide with or without a single phosphorylation PTM. And here you see basically the difference very clearly distinguished. So yes, we can measure that. And not only that, here we can also do things that mass spec cannot do. For example, here you see two serines in a peptide, where the first and the second one can be both not phosphorylated, or one phosphorylated, or the other, or both, right? And you see the four variants very clearly distinguished here. And say if you have the first or the second, so you have the positional information of which one is PTM, which has a PTM, and that you cannot get by mass spectrometry by principle, because it has the same charge and length in both cases. Again, we get very high single read accuracy. So this is an example where we are in the process of developing protein sequencing, and I would say de novo sequencing is still far out, but identification, so reference sequencing, like fingerprinting, that's already very, very possible. And there are companies now very close to releasing commercial products to do that for you, like Portal or Oxford Nanopore Technologies. So we can do that. Okay, so in the last part, the last 10 minutes or 8 minutes or something like that, if that's okay, David, yes? I will say something about building. Because, the last decades, we have used single molecule technology, nanotechnology, to study so much DNA-protein interactions, so many issues in biological sciences. But can we also start building life up from the bottom, right? I mean, some biologists would call me naive, but I like it. We have living objects like a cell, but if you would shake this up, you have the same components in a test tube. This is clearly not alive. This doesn't work. This does work. So what is it in the arrangements of atoms in time and space that make that work? So what are the fundamental minimal requirements to make objects a living object? So what is life, if you want to say it in a very general sense? Well, we know all kind of properties of living systems, like life can sustain itself in homeostasis, which is already very, very, very non-trivial. It can grow and divide, can sense and respond, it can evolve, and all these properties that non-living objects don't have, that combination. There have been, of course, many efforts so far already. There's famous efforts from Craig Venter, for example, who took mycoplasma cells and stripped those, the genomes to the bare minimum, and he found the bare minimum of sort of like 470 genes. And yeah, okay, I'm a physicist, 470 is a lot, right? I count one too many or something. 470 is like discouraging much. But that's sort of what he found. And so if you think about the boundary between life and non-life, basically he hits a barrier of getting simpler. In the Netherlands, in Europe, in Germany, and many other places now as well, US, Asia, there's a sort of more a bottom-up approach. Can we start sort of huge in vitro reconstitution, where we just bring molecules together and build with these molecules, and we have some big consortia. Now, there the great advantage, of course, is you know what you put in. You know exactly, you put, I don't know, tens of components in, you know what they are, right? But how to integrate all that into functional units? We take a bit of a modular approach in our research efforts. We have a big national consortium which I'm leading, where some people are doing metabolic units to try to build ATP homeostasis. Other people work on cell division, other people work on synthetic genome. And then we will try to integrate that, which is actually quite a challenge. But now we have a new project called Evolve, and okay, this was the selling slide for that thing, and so we will cross the gap between non-live and live, whatever. But we will actually also really use active learning to help us survey the parameter space, because it's so huge, and so rational design from the bottom up is actually hitting some barriers. So the basic approach there is that with a large group of 30 PIs in the Netherlands, we're trying to put molecules in vesicles and then through a number of evolutionary cycles to try to build functionalities of living systems in a living object. It's a 10-year program with a lot of funding there. Of course, that is in the end then actual research. This was more like the selling managerial overview of the project. In my own lab, I'm a physicist. I don't know anything about metabolism. It's too difficult for me. But we can do things with forces and shapes and structures and things like that. So my own efforts are mostly focused on cell division. And here you have if you have an object like a cell, you want to basically break the symmetry and then deform that object along that symmetry axis, and then in the end split it by abscission into two daughter cells. That's the basic phenomena of cell division. And we're studying all these aspects to some extent. So, for example, we developed techniques to generate vesicles, liposomes on chip. So we have this, what we call OLA octanol-assisted liposome assembly, where we have a multi-fork structure that forms these kind of things in this spontaneously split of octanol droppers, and then we left this beautiful liposomes. We can even mechanically split these by running into a sort of triangular shaped blockades thing, but that's cheating because it's possible, but it's very fragile. It needs very fine-tuning of conditions to do that. We have we studied the symmetry breaking, most famously done by Min proteins. Here you see an example of Min proteins in nanofabricated chambers. You can do it also in vesicles. There's also lots of work by Peter Svile in this area. We can put filaments in vesicles. Here you see some FDSE filaments. And then we studied, for example, in actual cells with Seamus Holden and Ethan Garner, some of the work that these FDSE filaments are sort of treadmilling around the circumference, and they're basically associated with a shrinking ring of this FDSE, associated with the deformation of these cells. And we can do this final stage of abscission. And just in the 3 minutes that are left in my talk, I will give you an example of a recent work that we did in this area. And this relates to a protein called dynamin A, which is involved in bacterial sporulation in Streptomyces organisms. And there was a previous work from Mark Brankamp on this, who worked actually on fusion rather than fission. So basically he put this dynamin A and had a GUV and SUVs, a small uniluminal vesicles and a giant luminal vesicles, and saw this fusion process being promoted by dynamin A proteins. And there was a speculation in the literature that for the same reason, because this is so-called forward topology, if you switch that around, you have a different topology. If you don't put the dynamin A on the outside, but on the inside, in this topology, it might actually then induce basically the closure of these membranes here, and therefore inducing cell division. This was not shown experimentally, I mean, that's what we've shown now in our own work. For that, there's work by postdoc Nicola De Franceschi. He did some deformations of liposomes. He first developed a technique to deform these liposomes. That's done by some DNA crosses which are cholesterol anchored. And by the cholesterol anchor, they induce curvature here. And in doing that, if you put it on the outside, then you basically make this kind of dumbbell-shaped structures. If you put it on the inside, by the way, you get this inverse stomatocyte structures like that. Okay, we use this kind of structures as a starting point for our cell division study. So we use these kind of structures. Here you see an experimental example, here you see another example. And now we do this where we encapsulate in our technique inside some dynamin A proteins. Okay, and if you do that, here's what we find. The dynamin A basically assembles at the neck. So it's a curvature sensing protein that assembles at the necks of these dumbbells. So you can see a single example here. You see this whole series of stuff here, which is really nice. And now we basically start probing what it does there. And we do this by FREP experiments. So basically in the FREP experiment, we FREP half of the membrane fluorophores of this kind of thing. And then over time, this grows back because fluorophores in this membrane can flow across that junction to the other side, right? If it's a connected structure like this dumbbell. All right, but what we found is that we had different classes of this kind of experiments. One is indeed what I showed you. We FREP half of this structure, and we see it come back because the fluorophores in the membrane flow to the other side of the dumbbell. But sometimes we would actually find that if we FREP this experiment, nothing comes back there. And sometimes we find something intermediate. I mean, it sounds a bit silly, but there's really three distinct classes there. So we actually interpret this as being, this is actually an open neck, where the dynamin A didn't do anything. Here actually it induced hemiscission, and this is full scission. In hemiscission, you basically fuse these particular lipid membranes, but not yet these ones. So you get this particular arrangement. And if you have that, indeed, the fluorophores on the outer leaflet would actually flow into the other part, but on the inner leaflet, they cannot. So you get a partial recovery in the FREP. Anyway, we did the statistics on that, and in our published data, we find actually that with dynamin A, we get a lot of this hemiscission and full scission. And actually, these numbers actually with follow-up work have improved to much higher numbers as well. So with dynamin A, we get actually scission there. Now, if you think about that, that's quite a thing, because now we have actually a single protein system that can work as a scission machinery here. Say, at least from a synthetic cell perspective, that's very attractive, right? Because there's, in biology, we know lots of systems like the escort systems or the dynamin A systems, the bacterial dynamin A's, the human there's the snare proteins and all that. And they work in these two different geometries, from the inside, from the outside, in forward and reverse topology. But this complicated slide is basically zooming in on this particular thing where we put these dynamin A's on the inside of these dumbbell-shaped structures. And in doing so, they then establish a division system. This is very exciting. So, summing up this third part of my talk, I showed you some example where we start building these biological molecules rather than only studying biological molecules. It's another theme is in nanotechnology where you can image, you can study the properties, and you can build with these kind of structures. So with that, I come to the end of my talk. I showed you some work of recent work on SMC motor proteins, some nanopore single molecule fingerprinting and sequencing, and some earlier data on synthetic cell division. And I want to give credit to my wonderful group members. And I do want to mention that we have no less than 5 postdoc openings. So if you happen to have an interest, please contact me and let me know. Thank you very much for your attention.
I'm sure there will be more than a few questions. Ken?
So on your telomeric barrier on the SMC, is it specific for telomeric proteins, or if you put arrays of other proteins?
It's not specific for the telomeric proteins, because we showed, for example, with LAC-I arrays, if you make a long, dense array of LAC-I, which doesn't have any telomeric function at all, it also works. So actually the telomeres prevent condensing passage and therefore slipping off and therefore lack of condensation of DNA near telomeres by just a physical mechanism, by having a physical dense long array.
It's just amazing, both the breadth and the depth that you're able to cover. An interesting point that you made that I wasn't aware of is that the main limitation for protein sequencing would be that A, you're sensitive for a stretch that's about 10 amino acids long.
Yes.
And you have a large alphabet. with really basic.
Yes.
So,
Can I briefly explain it to the audience, because we discussed this in the coffee break. But the idea is that in a nanopore sequencing, you have that nanopore. It's probing, say, a DNA molecule there, right? It is not one base there in, say, DNA sequencing. One base setting the current. It's actually a stretch of, say, 5 bases there. So the stick, and that's due to a geometric spreading resistance and thermal fluctuations. That combination makes that you have so-called K-mere of 5 bases. Now, single-strand DNA bases are separated by 7 angstroms. The amino acid by 3.5 angstroms. So you have 10 amino acids there. At every point, you have 20 possibilities, so it's impossible to build a library. Now your question.
Yeah, so you already answered it, yeah, right?
Oh, okay.
So with a biological core, somehow I might have expected that you could focus in even more. But the thermal
We're trying that. So yeah, we'll see where we end up. But I'm actually a bit pessimistic because it's such an intrinsic property. We worked with graphene pores in the past, for example, which would be the ultimate geometry, right? And they have their own material science property, but from a geometry point of view, that will be the ultimate low geometric spreading resistance and all that. We'll see where we end up. We are doing some protein engineering to make it even sharper. But, yeah. Better approach might be actually to make recognition groups there that binds specifically to, say, one of the 20 amino acids or a group of the amino acids, and then have combinations of that. That might work.
Just an immediate follow-up to that. How do you think the thermal fluctuations would change with some of these PTMs in there, given that some PTMs can be very, very large? So would that reduce your sensitivity?
The sensitivity of the nanopores is not really the problem, because they're hugely sensitive. This is more a geometrical problem. If you have like a local PTM at one spot along your amino acid chain, your peptide chain, it will fluctuate, but it doesn't matter because it will fluctuate on the nanosecond, microsecond timescale, and that you measure on the millisecond timescale, so those are averaged out to one particular current value. Is that answering your question? Okay, thank you.
Also following up on that, so the length of the amino acid chain is limited by the distance between the narrow neck and the helicase. Do you have plans to displace the helicase?
John is completely right. This approach that I showed you today is completely limited to a peptide length of, say, 25 amino acids. That's not very impressive because most proteins are hundreds of amino acids long, so you cannot do a full protein. Multiple things to say about it. One is there's already huge classes of important peptides, say immunopeptides, which are only 10, 15 amino acids long, which are on the outside of cancer cells, which are very interesting to measure, and there's very limited techniques to do that. So for that, there are applications that are very useful for that. The other thing is you can take the shredding approach, which people already use in mass spec, to actually take a digestion and then look at the digested fragments. We're looking into that. The third thing is actually, yes, there are approaches where you get rid of the helicase and use other modes to drive a complete protein through. With electroosmotic flows, we can drive probably mixed charged amino acid chains through proteins. And then by choosing conditions, we may be able to slow it down enough to do full proteins. So there's still, this is a rapidly developing and very young field. We have to see where we end up there.
So just follow-up question, out of curiosity, can you use AAA helicase, for example, that can thread protein through core?
Keepbacks.
Just AAA helicase, ATPases, that can, instead of threading nucleic acid helicase, you can thread.
Oh, yeah, sorry, that's a good point. Yes, it's possible to have a peptide translocase, basically. That's what you mean, right? I would be super interested to hear what kind of one you have in mind. There's a Clip X, which is the major one. People do use it. It doesn't make a step of a single amino acid per cycle, and that's really a disadvantage. Early data showed that actually the step size is 3, 4, 5, and rather random amino acids per step, which is really a disadvantage. More recent data from Jet Nivala at Seattle suggests that the step size is 2 amino acids. Still not 1, but fingerprinting can be done, and for example, indeed, the continuous translocation can be done that way. So he's working on that together with Oxford Nanopore Technologies.
I was also very interested in that sequencing example you showed. I noticed that you have all DDEE peptide. What happens if you have a peptide that is rich in hydrophobic amino acids?
Yes, I was very brief on that. In the very first measurements, we cheated, or we made our own life a little bit easier by only having a negatively tailed peptide, because that would be drawn into the nanopore just like the DNA. In more recent work, we work for more of these mixed valence peptides, so partly positive, partly negative. Those are problematic because the positive ones don't drag through the nanopore in the same way as the negative ones, actually go in the opposite way. So we're developing schemes, these electro-osmotic flows, where you basically coat, for example, the nanopore walls with a certain charge, and then in a field, this charge, the counter ions actually run through the nanopore and drag along the water and drag along the analytes, irrespective of the charge. That sounds almost like science fiction, but it can work, as is shown in a different layout of nanopore work. And some people have done mutations of the nanopore, so you can work with origami, or you can work with all kind of tricks to make the local charge there differently.
So that would something like that work also for hydrophobic coating?
Another point indeed, there's also hydrophobic, we found out very early on. So the bottom line is these nanopore signals, the nanopores are so sensitive, you get a very distinct signature in your current trace. Now the next question is, how do you interpret that particular wiggle? But that's where it becomes very specific to the particular peptide, and all these effects come into play. The particular residence of charges near the nanopore mouth, also hydrophobic interactions to the inner side of the nanopore, pulling the whole peptide aside, making larger currents, for example, counterintuitively. All these kind of things can be modeled by molecular dynamic simulations, but these are, of course, quite extensive for a particular peptide. So, yes, it's still a long way for de novo sequencing and simple biophysical interpretation of these signals. But the sensitivity is such that you can already use, as an engineer, these wiggles to identify with reference sequencing some peptides.
Right, in the interest of time, I'd like to thank Cees Dekker one more time.